Hello, my name is Carmen White, and I'm going to give you facts about DNA. DNA, what in the world is DNA? Deoxyribonucleics, very hard word, and also known as DNA, is what makes people who they are. I am credible to speak about this topic because I did the research to speak about it. I understand it and it makes sense to me being in the field that I want to pursue my career in. I want to be a lawyer, and in this field, DNA is something that is very crucial to whether someone is guilty or not guilty. This is important because before DNA analysis, many people went to prison for crimes that they did not commit. And we all know that that is horrible. I will speak about the facts regarding DNA. I will also mention research from Rada Saad from the Department of Pathology from the Bayer University Medical Center. After, I will discuss Stevenson Swanson from the Tribune National Correspondent and what he talks about of DNA. Then I will talk about the steps to DNA analysis. Then I will bring up the steps talked about from the National Institute of Justice. DNA are cells in the body that have no alike cells as another. This sets us apart from anyone else in the world. According to Rana Saad from the Department of Pathology from Baylor University Medical Center, historically, identity testing is the forensic field started with the analysis of the ABO blood group system. Later, new makers for identity and paternity identification were based on variations of serum proteins and red blood cell enzymes. Eventually, the human leukocyte antigen system was used. It was not until 20 years ago that Sir Alec Jeffreys, professor and geneticist at the University of Leicester in the United Kingdom, pioneered DNA-based identity testing. So it took a long time before DNA testing was even something that was even thought about or used in acts of crime or investigations for crimes that were committed. According to Mr. Swanson from the Tribune National Correspondent, the genetic material that determines an individual's physical characteristics is DNA. DNA makes up 3 billion pairs of molecules in the sequence unique to each person. That means no one has the same DNA alike from each other. Even if you are siblings, yes, you might have some DNA that matches or whatever due to your parents, due to you having the same parents and things like that. But your DNA is going to be apart from your siblings. He also states that this is incomparable means of identification. Base has developed astonishingly quickly since it was the first used as a forensic tool in 1985. By 1996, forensic experts were able to identify tissue and bone fragments from everyone on board TWA Flight 800, which exploded in the air off Long Island, despite the fact that there were no remains were recovered from the ocean floor. I'm sorry. That many remains were recovered from the ocean floor. The DNA process involves several key steps. Replication. DNA replication is the process by which a cell makes an identical copy of its DNA. This process is crucial for cell division, growth, and repair. During replication, the DNA molecule unwinds and unzips. With the help of enzymes like helicase, each strand serves as a template for the synthesis of a new complementary strand, resulting in two identical DNA molecules. Then you move on to transcription. Transcription is the process by which the genetic information encoded in DNA is used to synthesize RNA, which is ribonucleic acid. This process occurs in the cell molecules. RNA polymerase, along with other transcription factors, binds to a specific region of DNA called the promoter. It then reads the DNA sequence and synthesizes a complementary RNA molecule. The RNA molecule formed during transcription is called messenger RNA, which carries the genetic information from the DNA to the ribosomes in the cytoplasm. Then you move on to translation. Translation is the process by which the genetic information carried by the RNA is used to synthesize proteins. It occurs in the cytoplasm, specifically on ribosomes. The RNA molecules carries the genetic code in the form of codons, which are sequences of 3 molecules. Each codon corresponds to a specific amino acid or serves as a start or stop signal for protein synthesis. Transfer RNA molecules bind to specific amino acids and recognize the codons on the RNA through their anticodon sequences. As the ribosomes move along the RNA, it catalyzes the formation of peptide bonds between the amino acids carried by the RNA molecules, resulting in the synthesis of polypeptide chain. Then you move on to gene expression. Gene expression refers to the process by which the information encoded in a gene is used to synthesize a functional gene product such as protein. It involves both transcription and translation. The regulation of gene expression is crucial for controlling the types and amounts of proteins produced by a cell, which in turn determines its characteristics and functions. While I just explained the medical terms for DNA process and the long information about it, the National Institute broke it down in 3 simple steps. According to the National Institute of Justice, the process includes biology, technology, and genetics. The DNA is taken from a crime scene and taken off of whatever material or object that a person who committed a crime may have touched during the crime. The DNA is removed off of those items, and then it is processed to see how much of the DNA can be used to investigate. Then it is taken over to the computer lab or to the labs, where they're put through a system and put through a system process where they find PCR and then put into categories to be examined. Those are in a lab, and the DNA is sampled genotyping. When they go through that, they get after they get a certain amount of PCR, they go through a system, or going through the system, they then send it over to genetics. And in the genetic process, you find matches. And if you don't find matches, then you basically get rid of your so-called suspect or a potential suspect. So when it goes through genetics, they may have somebody else's DNA, and the DNA that they found at the crime scene may not match with the person's DNA that they took or that they sampled. So through that process is when they find out if a suspect was at the crime of the scene, or if the suspect was not at the crime of the scene. DNA is what makes you, and what makes me, me. It is what will make or break a case in an investigation. There are steps to DNA, and without the steps I mentioned, it is a possibility that your DNA may be compromised and inadmissible. If you don't do it correctly, then you might be putting somebody behind bars that should not be behind bars. These are the facts about DNA. I hope you all enjoyed the information. And remember, your DNA is the only DNA in this world. And I would suggest you not do the crime unless you're ready to do the time.